Hi everyone. Pretty cool, right? No, not the glass, but the act of reaching out for a glass of water. I'm thinking a lot of you are still very confused. But do you know that this task is actually quite complex? This so-called simple task is so complex that scientists and engineers to date have not been able to engineer a robot that's as good at this task as we humans are. To put things a little bit more in perspective, let me narrate the story of my 10-month-old niece, Anya Baby. So, Anya Baby's favorite pastime is trying to throw her toys over the fence of her playpen. And one day, she decided to take on a task as huge as her. Literally, she tried to put the pillow over the fence of her playpen. The first few times she's trying it, she's not quite able to grasp the pillow properly. She keeps on trying and she's finally able to grasp the pillow and raise it some way, but loses her grip midway. Being the perseverant little munchkin that she is, she keeps on trying and finally she's successful at the task. The reason why I narrated this tiny anecdote to you is so that you can remember that at some point in all our childhoods, we have quite extensively trained so that we can make the task of reaching out for a glass of water so effortless. My name is Riya, and I'm really curious to understand how our brain is so good at performing fine motor movements and learning new motor skills. So the region of our brain that's concerned with these functions is called the cerebellum, and it's located somewhere in the behind part, and it's the region that's highlighted in red in the picture. And I'm interested in understanding how the cerebellum learns. Now, when you think of learning a new motor task, for example, throwing a basketball through the hoop, you know that the first few times you try this, you'll miss the basket or the target by a lot. But if you keep on trying, you get better with time. You might even get so good that you can make it into the NBA team. So, how is it that we get better with time? The way our cerebellum does this is by actually getting feedback of our motor movement. So the feedback about the accuracy of your movement goes back to the cerebellum and is used to make your movement more smoother or accurate the next time. Before diving into details on what happens exactly in the cerebellum, let me remind you that neurons in our brain communicate through the language of electrical signals. So in the cerebellum, there's this interesting looking neuron called the Purkinje neuron. Pretty cool, right? It almost looks like how our hairs grew out during the pandemic. So this Purkinje neuron receives input from a group of cells called the granule cells, and it processes this information and sends it to other regions to finally generate a motor movement. Now, the function of the cerebellum here is to make your movement as smooth and accurate as possible. But when you start learning a motor task, the cerebellum is not very good at estimating how accurate or how good the movement would be. And you'll probably miss the target by a lot. Now, the feedback about the inaccuracy of your movement comes back to your cerebellum, back to your Purkinje neuron, in the form of electrical signals via a separate pathway called the climbing fiber pathway. This feedback is then used by the Purkinje neurons to alter the connections from the granule cells. So if you remember, the information or electrical signals that comes in from the granule cells is actually what is used to control the accuracy or smoothness of your movement. So refining or altering this connection would help to make that movement much smoother and more accurate. Now, if you can imagine, with trial by trial, your connections from the granule cells to the Purkinje neuron is getting more and more refined so that the movement gets more and more accurate with trial, and you get better with time. Now, when you think of making that three-pointer shot, you might know that it's not just about getting the ball through the hoop. You're actually grasping the ball a particular way. You have to make a jump, then you have to actually throw the ball and get it through the hoop. Then you have to land back on your feet and not lose your balance. So there's a feedback that comes back to the cerebellum regarding each of these tasks. So what makes things a little bit more complicated is that all these feedbacks come back to your cerebellum, back to your Purkinje neuron, not at the same time, but at different delays. So for example, the feedback of whether you landed back on your feet and were able to maintain your balance comes in almost instantaneously, but the feedback of whether the ball went through the hoop comes in at a considerable delay. How is it that the same type of neurons, that's the Purkinje neuron, is able to process all these different feedbacks that's coming in at different delays? The way we think the cerebellum is able to deal with this is by having separate sets of Purkinje neurons assigned for each of these feedbacks that's coming in at different delays. So there's a set of Purkinje neurons for feedback regarding whether you were able to maintain your balance, and there's a separate set of Purkinje neuron for feedback regarding whether the ball actually went through the hoop. Now, what is interesting is that the only difference between these neurons, the Purkinje neurons, is actually in the expression of a few molecules. How is it that the difference in expression of a few molecules enable these Purkinje neurons to process information differently? This is exactly what our lab is interested in understanding. And we try to address this problem using the mouse brain cerebellum. So what I mean by molecules is actually intracellular components in the Purkinje neuron that is involved in receiving information in the form of electrical signals from the granule cells, processing this information through the chemical pathways, and then finally generating an output and transducing it back to electrical signals, which is the language of the neurons. So a peek into the work I do in my lab would look like taking mouse brain cerebellum, making live cerebellar slices. Then I take these live cerebellar slices under the microscope, and I'm actually able to view the Purkinje neuron. Then I approach the Purkinje neuron with an electrode so that I can record the electrical signals that is emitted by these Purkinje neurons, which is the information that the Purkinje neuron receives or transmits. And then to understand how the differences in molecules enable these Purkinje neurons to process information differentially, I block the function of these molecules and see how the processing of the information gets affected. Now, I think that trying to understand how our brain learns a new motor task or makes a fine motor movement is quite interesting. But this is not the only reason that we study the cerebellum. The cerebellum is also involved in a lot of neurological disorders. For example, ataxia, a lack of coordination in voluntary movements such as picking up an object or walking can be caused by defects in the cerebellum. Now, interestingly, so far I have talked to you about the role of the cerebellum in motor functions. But do you know that cerebellum is also involved in emotional and cognitive functions? Many studies point to the contribution or role of cerebellum in autism spectrum disorders. Now, to understand the cause and effect of malfunctions of the cerebellum in these disorders, we need to actually understand how the cerebellum functions. In our lab, for doing this, we go all the way from the behavior level down to the molecular level. Here, I'll conclude my talk, and I hope that the next time you reach out for a glass of water, you'll be able to smirk at those robots who have not got it right yet. Thank you.